Today topic of discussion is T cells or T lymphocytes. First we will discuss that locations. T cells develop from hematopoietic stem cells in the bone marrow. So development that means origination from bone marrow, but it is usual development in the place of thymus. So T stand for thymus. T cells, depending upon the place where it used to mature itself, we used to call it as T cells. Because of it generate in bone marrow, but it is migrate to thymus for its maturation. So this one generated from bone marrow, but migrated to thymus for its maturations. Next point, that is the surface marker or identification marker. They are mainly regarded as identification marker CD3. If the cell is having in the help of experiment process, if we are seeing there is presence of CD3 marker, that particular XYZ cell, that means it is from the family of T cells or T lymphocytes. If we want to talk about that category, it divided into three types. First one is T helper cell, TH cell or T helper cell. Second one is cytotoxic T lymphocytes, TC cell. Third one is T regulatory cells. So three category: T helper cells, TH cell, TC cytotoxic T lymphocytes, then T regulatory cells. Further, it have co-receptor on its surface. TH cell having CD4 plus cell, TC cell having CD8 plus cells. These two are act as a co-receptor. There is one mandatory condition also. It is not going to encounter any antigen by itself. That means it need a combination. So these cells need a combination, or it need a interaction with some other cells. That means if a antigen present solely without the representation or without the presentation of representation, that time the T cell will not identify antigen, but the antigen is represented with the help of MHC molecules, then the T cells would be able to identify that antigen. For that identification, T cells need antigen presentation with the help of MHC molecules. But antigen present without MHC molecules, T cell would not able to attack. That's why there is no strong response towards that antigens. That's why the T cells can identify the antigen with MHC molecules only. So this is one mandatory condition that T cells will work presence of MHC complex only. The T cell will work presence of MHC complex only. There is absence of MHC, T cell not attack that particular antigen. So it provided strong response towards that antigen. So now solely focus on T cell co-receptor. Ultimately, we are going to see their action only during the encounter of any kind of antigens. Suppose this one is the surface of B cell. This one is the surface of the B cells. In general, whole surface molecule act as a co-receptor. These are the things act as a co-receptor. It may be T helper cells or TC cells. First, discuss about surface marker, that name. of the surface marker CD3. If it is a TH cell, it having co-receptor which is will be known as CD4 cell. TH cell, T helper cell having co-receptor CD4. CD4 having totally 4 domain it have. It have a single polypeptide chain only. This will be known as CD4 plus receptor. This is CD4 in the case of T helper cells. Next, there is a generalized receptor TCR, T cell receptor. Third one is TCR. It having two polypeptide chain together, it is known as TCR. TCR will going to interact with the MHC molecule on the surface antigen presenting cell whenever there is any encounter of antigen. In the case of CD8 plus cells, TC cells, that time there will be presence of second co-receptor. Next co-receptor that will be known as CD8 plus. It may be known as homodimer or heterodimer. First one name is called as alpha, second one name is called as alpha or beta. So this will be CD8 plus. Next, some other molecules are also there which are helping co-stimulation. Some other molecules also responsible for co-stimulation. That means if there are interacting towards containing interaction between two immune cells or between antigens, that time those molecules will be helping immune in strong response between those two immune cells. The name of that co-stimulator, first one is CD40L, second one is CD28. These two surface molecule, it will be helping in the co-stimulation process. Next, again some addition molecules. This one also helping interaction. Name of that addition molecule CD2 and LFA1. These are acting as a addition molecules. If the cell is T helper cells, there will be presence of CD4 only, absence of CD8. Suppose that cell is TC cell, means co-receptor CD8 only, absence of CD4. But here mention general. So all the cell drawn but normally CD4 or CD8 cell. If T helper cell means CD4 only present. TC cell means CD8 cell only present. Absence of CD4 cells. So whenever there is encounter of any kind of antigen, what is going to happen? Suppose there is one more cell. So that is any kind of immune cells, that surface will be MHC molecules. It is surface having MHC molecules. It will representing antigen on its surface. This TCR will interact with MHC molecules, and the cell also containing different receptor. So this cell containing different receptor. On its surface marker which is interacted with the co-stimulator and addition molecules. It will interacted with the co-stimulation, that co-stimulator or addition molecules. Enhance interaction between these two immune cells will be more stronger. That will be helping in the successful elimination of those particular antigen which is there in the body. So this way whole process will work. Locations: hematopoietic stem cells, maturation thymus. T stand for thymus cells. Identification markers CD3. Three category: T helper, TC cytotoxic and regulatory cells, co-receptor CD4 plus and CD8 plus. Here mentioning different type of co-receptor, but only one only presented. Along with co-stimulation and addition molecules, it will giving strong immunity against that particular antigens. So first one is surface marker CD3, second one is T helper cells, co-receptor CD4, it having 4 domain. Third one is T cell receptor alpha and beta chain. Then CD8 cell and R TC cells, CD8 plus is the co-receptor. Then co-stimulation CD40L, CD28. Then addition molecules CD2, LFA1. So any one kind of molecules only presented, surface marker having, but along with the addition and co-stimulator presented, it provided strong immunity. Successful elimination of those antigens, elimination of those antigen in the body. These are the way whole process will work. Thanks for watching.